Good morning. We are back with screencast number 3, a relatively short screencast, which is going to be covering DNA replication. Okay, and it is covered in chapter 25 in your book. We're going to cover it big picture. If you would like more depth, not only should you listen to the Amoeba Sisters, but they'll provide more depth there for those people who love that stuff. All right, so let's go through. All right, so first thing you should know here, first we're going to go over the process, and then we'll go over, as you see on the bottom with the colon, the enzymes. All right, so DNA replication takes place during the S phase of the cell cycle, G1, S phase, G2, in both mitosis and meiosis. All right, so as you know, before replication begins, the two strands that make up the DNA are held together by hydrogen bonds. Well, those hydrogen bonds are weak. Okay, they are not strong bonds. Hydrogen bond is a notoriously weak kind of bond, as you'll get in a couple of years when you take chemistry and maybe physics next year. Weak kind of bonds, all right? So what happens here is the following. There is an enzyme. An enzyme splits open DNA. It splits open DNA at a position on the DNA that's called a replication fork, which is right there on this strand right here. So basically a replication fork. So an enzyme, I'm going to make a larger. All right, there's your black enzyme right there on the replication fork. And by doing that, it's going to break the hydrogen bond, the 2 bonds or the 3 bonds, depending on which 2 bases you have together, therefore separating the strands. New and complementary DNA nucleotides, which are always present in the nucleus, fit into place by the complex of the complementary base pairing. All right, so these are positioned and then joined by another enzyme, okay, by another enzyme. And to complete replication, an enzyme seals any breaks in the sugar phosphate backbone. Remember the sugar phosphate, we'll get to the next panel. What I wanted to point you out here is this, all right, in DNA replication in eukaryotes, okay, each of the old strand, basically, if you look at the blue DNA separating here, that's the old parent DNA, becomes half of the DNA in the newly made strands of DNA. And that process is called semi-conservative. It's a semi-conservative molecule. And why? One could say this way, you're not continually making entirely new DNA. All right. I believe in prokaryotic cells, it would be totally conservative. All right. New DNA. But we don't worry about prokaryotic replication. Let's take a look at what you have going on here. All right. So the 3 enzymes that we want to go off, let's go over that process again. The enzyme helicase unwinds, unzips, opens, whatever word you want, breaks the weak hydrogen bonds at the replication fork, which would be right there in this model of DNA. All right, the nuclearly complementary base pairs are then positioned and joined by the enzyme DNA polymerase. So polymerase to build a molecule. We're making a DNA molecule there. All right, to complete replication, an enzyme called DNA ligase. So it is important to put that in because there is an RNA ligase too. So let's give you that much specificity. DNA ligase joins the molecules together. Helicase, polymerase, ligase. Okay, so there is so much if you take a look here at relationship to cancer treatments here. So replication. So for instance, as I read in your textbook recently, because I like to read that book, chemotherapy drugs for cancer treatment stop replication. That's their function. Okay, and then because it stops replication, it stops cell division. So that's the main job of a chemotherapy drug. There are a lot of different ones of those out there. All right, so that then would do it with a short stint on DNA replication. End screencast.